Alrighty, so now let's get into reviewing some of the fundamental concepts about genetics. So keep in mind that everything is made up of cells. There's two major types of cells, eukaryotic and prokaryotic. Prokaryotic cells tend to be the simple cells that only have a cell wall, a plasma membrane, and then they have regions of DNA that is contained within a chromosome, but it's not encapsulated by a nucleus, like in the eukaryotic cells. So eukaryotic cells are much more complex. They have many organelles, including the nucleus that is going to house the DNA. So the function of some of these organelles, more specifically the nucleus, its job is not only to contain the DNA, but is also going to protect that DNA from other organelles and other proteins that could degrade DNA over time. So the nucleus is one of its major functions is it controls access to DNA. So genes can only be turned on and turned off or go through transcription or translation by nucleic control, or the nucleic control. And the nucleus is going to help to keep the DNA separate from the metabolic machinery that is going to function 24 hours a day. So contained within that nucleus is DNA, deoxyribonucleic acid, and so we're gonna look at DNA pretty in-depthly as we go through the semester, but essentially DNA is going to provide the blueprint for all form and function of an individual's life. And DNA makes up chromosomes that are going to also reside within the nucleus. So DNA structure is double helix, meaning that two strands are going to be linked together with hydrogen bonds. And DNA is going to be built with subunits called nucleotide. And these nucleotides are composed of three major components: a ribosugar, a phosphate group, and a nitrogenous base. So the four nucleotides that make up DNA are going to be thymine, cytosine, adenine, and guanine, or A, G, C, and T. And these these nucleotides are going to be organized in DNA and create genetic code for proteins based on precise sequencing. The nucleotides are also going to vary, but it's very specific how they vary. So adenine is always going to pair with thymine, and guanine is going to always pair with cytosine. And we're going to study this more in depth, and you guys will have a better idea of why those nucleotides pair with each other and only each other later on in future chapters. So the DNA replication process happens a couple of different ways. So we make DNA every time a cell divides, and that is primarily going to happen in mitosis if we're talking about somatic cells. So just an overview of this process: the double helix is going to unwind, the new DNA is going to assemble or be synthesized off of the older template strands, and that older template strand is then going to create two new strands of DNA. So as we look at total genetic material contained within different species, we know humans have 46 total chromosomes, 23 pairs. Cattle have 60. A lot of our livestock species vary in between 50 and 60 chromosomes. And then our plant species can vary anywhere between 33, such as in bananas, or clear up to 90 chromosomes, as in sweet potatoes. So sweet potatoes are very complex, and they actually are going to have 45 total pairs of chromosomes. So DNA very much is the language of life, and I hope you guys can get a good feel for the sequence and how DNA, or where DNA actually exists at. So if we have a plant here that is tangible, that's what we're most familiar with. Within that plant is contained cells. Those cells make up the tissue that is associated with the plant. And then within those cells are going to be chromosomes. And those chromosomes are going to reside within the nucleus, which is kind of the center of the cell. And those chromosomes are going to be looped in a double helix fashion, and that is going to be made up of DNA. So our chromosomes are made up of DNA, and DNA is made up of the 4 nucleotides, A, G, C, and T. So the way that chromosomes actually are assembled in an organism is what we call a karyotype. So this is a human karyotype where we have 23 pairs of chromosomes, so 1 clear down to our sex chromosomes here, which are 23. And we actually have the ability to extract a sample of DNA, arrest DNA at a certain stage, and then be able to stain DNA so we know which one is chromosome 1, chromosome 2, etc. So karyotypes are done in women that are over 38 years of age, so that because that is the first year of age that women are more at risk of producing a child with some sort of learning disability, but more specifically a child with Down syndrome. So let's review some terminology. All right, so just to orient you to this figure we have here, we have 2 chromosomes. So you can think of both of these chromosomes as chromosome 1. So 1 from mom, 1 from dad. When you hear the term homologs, those are going to be chromosomes that are alike. So this purple chromosome is a homolog to this blue chromosome here. A gene is also going to be referred to as a loci. I use them interchangeably, so just be aware of that. And that's going to be the site of a gene on a chromosome. An allele is going to be one copy or an alternate copy of a gene. So for example, if we have chromosome 1 from dad and chromosome 1 from mom, and both of these chromosomes exist in a new offspring, within that chromosome contains a certain area or a certain position that's going to code for some sort of trait. Okay, so however, if we look down here towards the bottom, we may have a different gene that is going to be expressed. So maybe the gene that mom gave us is going to be expressed, and the gene that dad gave us is going to be suppressed. So that's an allele, is an alternate form of a gene. And then finally, gene pairs are where 2 alleles, one from mom and one from dad, are going to be referred to together. All right, so a gene is a specific segment of DNA that codes for a very specific trait. And and within a gene, it contains a promoter region. This promoter region is a start site where the gene is going to be turned on. So we're going to get really familiar with promoter regions when we get to chapter 13, which is transcription. Genes also contain introns, which are going to be these yellow areas, and exons, which are the pink areas. And these are going to be coding or non-coding regions. So realize that all the DNA material on a chromosome that's contained within a gene isn't necessarily all used to code for a specific trait. So within a gene, you may get certain areas like here in pink that are exons. These are going to be coding region where mRNA is going to be synthesized. And then you may have a break in information or an intron region where the DNA will not be synthesized into messenger RNA. So exons are going to be coding regions. They're going to produce mRNA. Introns are non-coding regions that traditionally have been thought of as junk DNA, but now we're actually finding that a lot of intron regions are coding for how that gene is regulated later on. So we'll talk about that when we get to the biotech section. All right, so the central dogma for trait expression is that we have a gene is going to code for a protein, and the protein is actually going to be the component that causes some sort of effect or the phenotype. Okay, so the proteins are going to express the genetic traits. Protein synthesis is synonymous to gene expression. So you will hear me use those two phrases interchangeably throughout the semester also. So when we think about this on a cell basis, genes are going to be turned on within the nucleus. The gene is then going to make a message called messenger RNA, and that messenger RNA is also going to be produced in the nucleus. But before messenger RNA can be synthesized or translated into protein, it has to be transferred out of the nucleus to the endoplasmic reticulum, where the protein is then going to be made. All right, so with transcription, this is the process of taking DNA and turning it into messenger RNA. So the first step is the gene is going to be turned on, DNA is going to unwind here at the start site, and then the RNA polymerase is going to come in and sit on the promoter region and transcribe or copy the gene. And the copy or the product is called messenger RNA or mRNA. When mRNA is made, it's also going to be made up of nucleotides, where G is going to pair with C, and A is going to pair with uracil, and uracil is going to replace thiamine. So if you see the nucleotide U anywhere, you know that you are dealing with messenger RNA and not DNA. Translation is the second process that takes mRNA and translates it into a protein. So mRNA is going to be read in a set of 3 nucleotides called a codon, and those codons are going to code for a specific amino acid like methionine or tyrosine, things that you learned about in chemistry. And then transfer RNA components are going to bring in the appropriate amino acid to the ribosome for protein synthesis. And we're going to look at both transcription and translation very in depth in chapters 13 and 15. All right, so, sorry, let's go back to that last slide really quickly. So keep in mind that a ribosome is going to be part of this translation process or transcription process, and the ribosome is going to have the ability to match the right transfer RNA with the right codon. And that transfer RNA is going to carry an anticodon to match the codon that is needed for the messenger RNA. And then all of these amino acids and codons are going to be paired together in a chain of amino acids. And it's the chain of amino acids that become the protein. So you've always heard that amino acids are the building block of proteins, and so now you can start to understand why. All right, so that is the end of chapter 1. And don't forget to do your reading quiz for chapter 2, and we will see you guys next week.